structure and functions of chloroplast genome. First, the introduction and brief history. Photosynthetic organisms contain independent chloroplast genomic DNA in addition to nuclear and mitochondrial genomes. Chloroplast is an essential cellular organelle found in the photosynthetic algae and plant cells. According to evolutionary perception, chloroplast evolved by endosymbiosis between non-photosynthetic protists and photosynthetic cyanobacteria. The origin of chloroplasts dates back to more than a billion years. The identification of Bengiomorpha, a fossil red alga which is aged 1.2 billion years, provides strong evidence to this point. Schimper is regarded as the first to have considered that plastids arose from endosymbiotic photosynthetic bacteria. He noted that plastids divide by binary fission independently of the surrounding cell and showed a remarkable resemblance with free-living cyanobacteria. Mirashchowski in 1905 termed the plastids as little green slaves working for their host cells to produce food from sunshine. Lynn Margulis in the late 1960s proposed endosymbiote theory, which emphasizes that symbiosis was a major force in the evolution of cells. The endosymbiotic origin of the chloroplast is emphasized by the relationships between these genes and their counterparts in bacteria. The organization of the ribosomal RNA genes in particular is closely related to that of a cyanobacterium, which pins down more precisely the last common ancestor between chloroplasts and bacteria. The general structure of chloroplast DNA. The chloroplasts are semi-autonomous organelles possessing their own genetic material. The chloroplast genome or plastome. First of all, Riss and Platt in 1962 reported DNA molecules in the chloroplast of the Chlamydomonas. Later on, DNA molecules were reported from the chloroplasts of other algae and higher plants. Chloroplast DNAs of higher plants are double-stranded molecules of relatively small size, ranging from 35 to 217 kilobases, with most of the photosynthetic organisms in the range of 115 to 165 kilobase. Only a proportion of the total chloroplast DNA is circular. They are present in 1,000 to 10,000 copies per cell. The molecular weight of chloroplast DNA commonly range from 85 to 140 into 10⁶ daltons, depending on the species of organism. The contour length is frequently around 45 micrometer, but they range from about 40 to 60 micrometer, depending on the species. The isolated chloroplast DNA typically exists in a variety of forms and conformations. For example, some are unicellular and others appear as interlocked dimers. There are two types of dimers: circular dimers, which are formed by a recombination between two monomers and may constitute about 10% of cpDNA, and catenated dimers, in which two monomers interlock like links in a chain. The catenated dimers may constitute up to 2.5% of cpDNA. The monomers may appear as relaxed, that is, open circular duplexes in vitro, but in situ, the closed, that is, supercoiled form is predominant. The first plant genome to be completely sequenced was a chloroplast genome owing to the relatively small size. In 1986, the chloroplast genomes of tobacco and the liverwort Markinsia polymorpha were sequenced, making these two the first complete plastome sequences. During the last 2 decades, plastome sequences have increased at a rapid rate, and currently 170 complete plastid genomes are available. These include plastomes from various groups of organisms ranging from green and red alga to land plants. The circular plastome of most land plants and algae have a quadripartite structure comprising of two identical or non-identical inverted repeats which separate two single copy regions, that is, the large and single copy regions LSC and SSC respectively. Size variation in the inverted repeats is the main cause for plastome size variation between different genera. The inverted repeat size averages around 20 to 30 kilobase, although exceptions are known. Genes located in the inverted repeats are, as expected, present as duplicates. The Gymnosperm pinus thunbergi has an extremely reduced pair of inverted repeats, sizing a mere 495 base pair, the smallest known among all plastomes, and in turn a small plastome of about 119 kilobase. This genus has lost all its genes coding for NADH dehydrogenases. On the other extreme is Pelargonium hotorum, which possesses inverted repeats of about 76 kilobase, giving its plastome a size of about 217 kilobase. Inverted repeat size variation is not the only cause for differences in the sizes of plastomes. Loss of genes, as in case of pinus, is another key reason. The lack of inverted repeats is thought to directly affect the nucleotide substitution rate in genes present in the remaining copy. Differences in the intergenic regions are also universal players in plastome size variation. The inverted repeats are thought to act as stabilizing regions and evolve 2 to 3 times more slowly as compared to the single copy regions. Plastomes are able to retain signatures of evolutionary history much better than their nuclear counterparts due to lack of recombination, which is in turn due to the presence of these inverted repeats. The large single copy region is the least conserved when all parts of the plastome are compared. The organization and structure of plastomes is generally stable and conserved. It does not mean that rearrangements have not occurred. There have been group-specific inversions, translocations, insertions, and deletions. A typical example is the inversion of about 50 kilobase segment in the large single copy region of legume plastome. The genes found in chloroplast DNAs are generally conserved with respect to content and order, especially within a particular group of organisms. These can be divided into 3 broad categories. The first category comprises of genes for the photosynthetic apparatus. This category includes photosystem first, photosystem second, cytochrome B6F, ribosco, then ATP synthase, and NADPH dehydrogenase genes. The second category comprises RNA genes and genes for the genetic apparatus. This includes transfer RNA, ribosomal RNA, RNA polymerase, and ribosomal subunit genes. The third category comprises of conserved open reading frames called YCFS, that is hypothetical chloroplast open reading frame, and potential protein coding genes like MAT, K, and SEM-A. The chloroplast genes are involved in 4 main types of processes, that is transcription, translation, photosynthesis, and the biosynthesis of small molecules such as amino acids, fatty acids, and pigments. The chloroplast genome codes for about 50 proteins including RNA polymerase and ribosomal proteins. Paradoxically, all of the known proteins encoded in the chloroplast are part of larger protein complexes that also contain 1 or more subunits encoded in the nucleus. The chloroplast genome codes for all the ribosomal RNA and transfer RNA species needed for protein synthesis. The ribosome includes 2 small ribosomal RNAs in addition to the major species. The transfer RNA set may include all of the necessary genes. The organelle genes are transcribed and translated by the apparatus of the organelle. ATGC content and RNA editing in plastid genome. Plastid genomes are highly AT rich, the average being around 63%. The plastomes of the moss Phycomitrilla patens, red algae Gracilaria tenuisitipetita, and the hornwort Anthoceras formosi have an AT content toward the higher end, that is 71.50, 70.90 and 67.10% respectively, while the green algae Nephroselmus olivacea has a low AT content of 57.90%. The AT content of the single copy regions is generally much higher than the inverted repeat regions. The low AT content of the inverted repeats is attributed to the presence of ribosomal RNA genes, which have the lowest AT content. The strong AT bias is also reflected in the codon usage. Roughly 70% of the codons of all plastomes end with the bases adenosine or thymine. Medicago, which has lost one of its inverted repeats, has an adenosine thymine content of 66.03%, which is comparable to other plastomes. Another feature which is common among plastomes is the conversion of bases at the transcript level, which can lead to a functional protein or a protein with a changed amino acid. For example, in hornwort Anthoceras, the fern adiatum or the monocot there are stop codons present in the coding regions of the plastomes, but it does not lead to a truncated transcript or protein. Instead, RNA editing affects a single base change in the messenger RNA that alters the codon, changing it from a non-sense codon to a sense or from one sense codon to another, which might lead to change in the amino acid. The usual alterations are C to U transitions, that is pyrimidine to pyrimidine in the transcript molecule. Plastid genomes of higher plants possess a number of open reading frames of unknown function. These conserved ORFS have been named YCFS, that is hypothetical chloroplast open reading frames. Some are specific to chloroplast genomes, some common to chloroplast and cyanobacteria, and some are found only in cyanobacteria and algal chloroplasts. Ribosomes of chloroplasts. The chloroplasts contain 70 S-type of ribosomes, which account for up to 50% of the total ribosomes in photosynthetic cells. The 70 S ribosomes of Euglena chloroplasts are found to require magnesium 2+ for their stability and also have a requirement for N-formyl methionine tRNA in chain initiation protein synthesis like the bacteria. The chloroplast ribosomes resemble those in prokaryotes in the features like use of N-formyl methionine as initiating amino acid, sensitivity to chloramphenicol, lincomycin and spectinomycin, which inhibit protein synthesis, and basic structures of their RNA and proteins. The ribosomes of the chloroplast consists of two ribosomal RNAs, that is 23S rRNA and 16S rRNA. Polyribosomes or polysomes have also been separated from the chloroplasts. The chloroplasts also contain amino acyl tRNAs, amino acyl tRNA synthetases, methionine tRNA. The presence of such ribosomes provides evidence that these organelles contain a specific protein synthesizing system. This protein synthetic mechanism of chloroplast is inhibited by chloramphenicol like that of mitochondria and bacteria. Chloroplast gene transfer. When the coding capacity and size of the chloroplast genome is compared to their predecessor, that is cyanobacteria, there is a vast difference. The chloroplasts have a much reduced genome as compared to their predecessor, but still retain many proteins not encoded by them. Chloroplast genomes encode only about 5 to 10% as many proteins as free-living cyanobacteria. This discrepancy is due to the fact that many of the genes have been transferred to the host nucleus, and a complex transport system has evolved where proteins required by the organelles are encoded by the host cell nucleus and then transported into the organelle, a phenomena known as the endosymbiotic gene transfer. This loss of autonomy from a free-living organism to an endosymbiote was a gradual process where duplicate copies of genes were generated and one of the copies was transferred to the nucleus. Thus, there was an intermediary stage where the gene was present in both the organelle and the nuclear genomes till the nuclear copy acquired suitable regulatory elements and became functional. The transferred organelle gene has two main obstacles to cross before the nuclear copy becomes functional, permitting the loss of the organelle copy. The first one is expression and the second is targeting. If the product of the organelle gene has to return back to the organelle, then the nuclear copy of the gene has to acquire suitable regulatory elements, organelle targeting signals, and transit peptides. The different possible mechanisms of gene transfer of the nucleus and targeting of the protein back to the chloroplast are: number 1, direct transfer of chloroplast gene after origin of introns and or editing, followed by gain of suitable regulatory elements and removal of introns in the nucleus. Number 2, direct transfer of chloroplast gene prior to origin of introns and editing with or without transient intermediates. And number 3, reverse transcription followed by integrative recombination of cDNA into the nuclear genome. Irrespective of the mode of transfer, the main requirement is integration and acquisition of suitable regulatory elements along with a transit peptide, which will lead to the transfer of the protein back to the chloroplast. Replication of chloroplasts. Chloroplasts arise from pre-existing chloroplasts during the life cycle of higher plants and are passed on to the progeny cells during cell division. The precise manner in which chloroplasts divide is not known, but electron microscopic evidence suggests that division is executed by a type of binary fission similar to that which occurs in bacteria and mitochondria. A constriction develops near the center of the plastid, and two progeny chloroplasts result from a pinching together of membranes in this region. Chloroplast division appears to be generally asynchronous in a given plant tissue or cell. However, a number of environmental factors including light, temperature, growth regulators, and minerals are known to influence replication and differentiation. Plastid inheritance. It is generally accepted that there are three modes of plastid inheritance in angiosperms, namely uniparental maternal, uniparental paternal, and biparental plastid inheritance. The uniparental maternal plastid inheritance is dominant in angiosperms. Its cytological events are characterized as the physical exclusion of plastids, the alteration of plastids during male cell development, and the exclusion of the male plastids during syngamy. The plastid alteration is mainly caused by the elimination of plastid DNA in the generative or sperm cells of the pollen. In the biparental plastid inheritance, the generative and sperm cells contain plastids and their DNA persistently, which are regularly transmitted into the zygote and in turn into the next generation. But the frequency of biparental plastid inheritance in certain cases may be influenced by control of nuclear genotype, the role of plastome in the process of plastid transmission and possibly in plastid multiplication, and the ratio of plastid number between male and female cells and the plastid distribution in the zygote. Recently, uniparental paternal plastid inheritance has been demonstrated in Medicago stava, Duckus, and Forbitus. These results indicated that this mode of plastid inheritance is definitely present in angiosperms. The cytological mechanism is that the plastids preserving their DNA in the generative and sperm cells may also be transmitted into the zygote. On the other hand, the female plastids are incapable of genetic transmission. This mechanism is concerned with the fact that the egg is highly vacuolated, in which most plastids are positioned in the micropolar end. In addition, the DNA replication rate is higher in the male plastids than in the female plastids within the zygote. The cytoplasmically inherited traits are identified by the following facts. Number first, the inheritance pattern in reciprocal crosses is different. The progeny shows only the characteristics of the female parent, and this is attributed to unequal cytoplasmic contributions of male and female parents. Number second, the absence of segregation at meiosis, or the presence of the type of segregation which do not follow Mendelian laws. Number third, persistence of a trait in the progeny after repeated back crossings when the backcross parent shows an alternative character. Number fourth, mutagens may cause 100% mutations in the plasma genes, and thus they differ in degree when compared with nuclear genes. Significance of chloroplast genome. The chloroplasts contain multiple copies of a DNA molecule, the plastome, that encodes many of the gene products required to perform photosynthesis. The plastome is replicated by nuclear encoded proteins, and its copy number seems to be highly regulated by the cell in a tissue-specific and developmental manner. Photosynthesis is a complex process that occurs in chloroplasts of higher plants and requires a large number of proteins to assemble the photosynthetic machinery. Many chloroplast localized proteins are nuclear encoded and must be imported into the chloroplast from the cytoplasm. A considerable number of genes for photosynthesis and other chloroplast functions, including transcription and translation, are encoded in the chloroplast genome, that is cpDNA. The plastid genome is a rich source of phylogenetic and other comparative data in plants, giving useful information about the relative age and relatedness of the organisms possessing them. Besides their use in phylogeny, these plastid genomes would help in rapid, accurate, and automated identification of species, providing plant taxonomists with an easy way to use species-specific DNA barcode catalog. The chloroplast genome transformation of higher plant has become the research hotspot of plant genetic engineering, with several advantages over nuclear genetic engineering, and it has become into a powerful approach for both basic and applied research. These applications include the studies of rearrangement of ribosome and the structure, transcription, translation, and RNA editing of genes in chloroplast, the production of antibodies, vaccines, poly-beta hydroxybutyrate, and bioelastic protein using chloroplast as bioreactor, the generating of transgenic plants with resistance to insect, disease, herbicide, and drought, and decreasing the gene flow of transgenic plants. Perhaps a complete understanding of the chloroplast genes and the products that they encode will have important practical applications in the future. The information of the exact mechanism by which Photosystem's first and second function might someday permit scientists and engineers to build a totally synthetic system capable of duplicating the capacity of green plants to capture light energy and convert it into chemical forms useful to living organisms.